Professor Warren Tate, or Emeritus Professor Warren Tate is here. Warren has been working in many research areas over the last almost
54 years.
Firstly in ribosome biology, but also more recently, relatively recently, in memory and also MECSF. CFS. And that's his topic for today. So it's my great pleasure to welcome Warren, and we're really looking forward to hearing your information.
Thank you. Well, thank you for the invitation to give this lecture today, and particularly to be able to talk about a neurological illness that really hasn't been in the mainstream of neuroscience, and that is myalgic encephalomyelitis / chronic fatigue syndrome. Amazingly, it was back in 1969 that it was designated a neurological disease by the World Health Organization, but there's been very little understanding of it, and I come into this lecture really with it as an unexplained disease. And what I want to tell you is what we've been doing to try and understand the biological basis of the disease. Now, why was it determined as a neurological disease? And firstly, I'd like to just talk about these outbreaks. There have been about 75 outbreaks of infectious disease since 1930, whereby after the outbreak has been resolving, people infected have had ongoing disease. For example, the first one, I've just listed the ones here during my lifetime. So I was born in 1946. And so the first one in Iceland, in a northern town in Iceland, I won't attempt to say what the disease was called, but that's the name of the town. About 1,090 cases. 40 years later, people have gone back to try and find the people that were infected and found about 80% of them still have serious ongoing disease from that original outbreak. And that's sort of typical of what's happened. The one at Royal Free Hospital in London in 1955 has had much more publicity, and there was about 292 cases at the hospital, mainly nurses, administrative staff, and a few doctors. And it was clearly quite a serious infectious disease because people were hospitalized all around in other hospitals in London. And again, that had ongoing disease. And that's where the name ME, myalgic encephalomyelitis, came from. Myalgic because pain was known to be associated with this ongoing disease, and cephalo because of the brain was implicated, and the itis because they felt there was inflammation involved. And then we had our moment of fame in New Zealand with Tapanui flu, where the local GP, Peter Snow, found a large number of people in the community coming with a fatigue disease. It's never been designated what it was. And 28 cases at least have been reported, and I don't know if there were more than that, but certainly in the annals of history. And then a year later in Incline Village in Nevada, small village with 2 GPs, they found that screeds of people were coming into their practices with severe fatigue. So they actually contacted the Centre for Disease Control and got them to come over, and they assessed the situation and wrote a report and determined that it was mass hysteria. But we now know these people have had ongoing disease, and this is where the chronic fatigue syndrome name came from. So in America, it's been referred to as chronic fatigue syndrome, from the UK, ME, and now the name that's been generally accepted really only just a few years ago is ME/CFS, and that's what I'll refer to for the rest of the talk. So the question that arises now with the pandemic going and reports of ongoing disease, which I hope to talk about at the end of the talk of this time, is this going to be another super spreader end of the ME/CFS. And so that's a question which is still to be answered. So why is this disease regarded as a neurological disease? Well, patients who are affected by the disease, this ongoing condition, have a number of neurological functions affected. For example, they have difficulty with homeostatic control, like temperature control, and their output of cortisol is low. So that is suggestive of hypothalamic dysfunction. They're very intolerant of any therapeutic drugs, such as simple painkillers or sleep medication or antidepressants. And I was giving a talk on Saturday at the AGM of the ME/CFS support group for Otago and Southland, and the chairperson came up and said what he finds most frustrating about the illness is orthostatic intolerance. When he shifts from sitting to a standing position, he feels dizzy and faint all of the time, and he's had the illness for over 40 years. And a classic symptom is cognitive dysfunction, and patients routinely call this brain fog. They can't concentrate, their memories become faulty, and cognitive abilities they normally have, they lose. And I'll say a little bit more about that later. Patients have frequent headaches, and they're very susceptible to getting migraines, and that may be because their sensory perceptions are much greater. So they have disturbances of perceptions of light, sound, touch, and smell, super sensitive to those compared with the rest of us. There's examples of many of them have neuropathic pain, which is stabbing and searing and frequent. Sleep dysfunction is one of the main symptoms, hyperinsomnia, and even when people with ME/CFS can sleep, they wake up with feeling totally unrefreshed. And the central symptom is pervasive fatigue, and the thought is that that's probably brain-related rather than just cellular-related. So how does one make a diagnosis of this disease? Well, I can say with difficulty at the moment. Generally, one has to have the symptoms and persistent fatigue for 6 months. And if a health practitioner has some knowledge and understanding of ME/CFS and has that in the possibilities, then what they generally do is to try and eliminate other mainstream illnesses which have fatigue as the main component before they make the diagnosis. But this is very difficult for health professionals and patients alike. Patients, because often suddenly they come down with all of these horrible symptoms, and so they go to their general practitioner to get some help, and the general practitioner doesn't have anything to offer, maybe because of a lack of knowledge. But even with the knowledge, there is over 20 clinical case definitions for ME/CFS in the literature, so it makes it very difficult for diagnosis. And there's no molecular diagnostic test. So when a practitioner orders a blood screen, generally it comes back normal or with just a few things around the fringes, nothing to suggest there's a major illness. Now in New Zealand and also elsewhere, there's been no exposure to this illness in medical school training. And this has been the case up until 2013, when I was asked if I would give a session in a module in third year medicine at Otago called Unexplained Diseases. And that tells you kind of where the knowledge of ME/CFS was in 2013 among kind of mainstream medicine. So if a diagnosis is made, what are the consequences for the patient? Well, sadly, this is a lifelong disease at the moment. And in 95% of cases, they don't actually recover completely from this illness. We do define now today two states, an acute state, and what that means is the person is housebound, maybe bedbound, and unfortunately 25% of the cases have that for life. Fortunately, 3/4 of people with the illness go into a chronic state where they're able to go out into society and do things, but they have a very restricted life. They suffer from the symptoms ongoing, and it can be very socially isolating. So what has all this uncertainty and unexplained nature of the disease meant for patients then in the past really 30, 40 years? And sadly, it's meant many of them have felt totally neglected. And I wanted to, on this slide, give you at least some good news. And Norway is, in fact, one country which seems to be been quite enlightened. I met a woman, and I'm sorry I've forgotten her name. I wish I'd remembered it. In London in 2017, who through family connection with the illness established a national association. And it seems like Norway has accepted this disease as real and is quite enlightened and is ahead of most of the rest of the countries. However, contrasting that in the United Kingdom, where a psychiatrist, incredibly influential, Sir Simon Westley, he has won awards, and as you can see, he's got a knighthood. He's been president of the Royal Society of Psychiatrists. He's focused his attention for a number of years on this illness and determined that it was a perception disorder, that he accepts the fact that might have been an initial organic cause, but actually the symptoms and the disability that patients have is a perceived disability and their perceived symptoms. And it's been called an opting out syndrome. Now, not surprisingly, in families that have been affected by this illness, this has caused great outrage. And for many years, there was a battle between him and holding his position and the families and groups that represented those families. Now, I call this a bit like a Trumpism. He's a very highly respected person. He's made these pronouncements, and unfortunately then when someone does this, as we see with Trump, it kind of goes on down below them and leads to some quite outrageous practice. And in the United Kingdom, in fact, young women were taken away from their families and put into psychiatric institutions, and many of them just withered away and some died. And a mother of one of these young women, Natalie Bolton, actually sent me a DVD of a documentary she had made. It's an award-winning documentary now with her and her son. And I'll just let you read that because it tells you not only the grief of the family, but the dark place, really, that ME/CFS patients were in for that period of time in the United Kingdom when essentially they were often being put into psychiatric institutions and not receiving very good care. What I'd like to do now is to let you hear from an ME/CFS patient herself. She's in her 20s, which is typical, and she's a woman, which is typical again. And I think she can best describe what ME is like rather than me actually giving you a long explanation. So this is Jennifer Bria.
Hi. Thank you. So 5 years ago, this was me. I was a PhD student at Harvard, and I loved to travel. I had just gotten engaged to marry the love of my life. I was 28, and like so many of us when we are in good health, I felt like I was invincible. Then one day I had a fever of 104.7 degrees. I probably should have gone to the doctor, but I'd never really been sick in my life. And I knew that usually if you have a virus, you stay home and you make some chicken soup, and in a few days, everything will be fine. But this time it wasn't fine. After the fever broke, for 3 weeks, I was so dizzy, I couldn't leave my house. I would walk straight into door frames. I had to hug the walls just to make it to the bathroom. That spring, I got infection after infection. And every time I went to the doctor, he said there was absolutely nothing wrong. He had his laboratory tests, which always came back normal. All I had were my symptoms, which I could describe, but no one else can see. I know it sounds silly, but you have to find a way to explain things like this to yourself. And so I thought maybe I was just aging. Maybe this is what it's like to be on the other side of 25. Then the neurological symptoms started. Sometimes I would find that I couldn't draw the right side of a circle. Other times I wouldn't be able to speak or move at all. I saw every kind of specialist: infectious disease doctors, rheumatologists, endocrinologists, cardiologists. I even saw a psychiatrist. My psychiatrist said, "It's clear you're really sick, but not with anything psychiatric. I hope they can find out what's wrong with you."
Jennifer Brett is a remarkable young woman. She made a documentary of her illness. In fact, she filmed herself during the years of her 3 or 4 years of her illness, staggering along corridors, trying to get to the loo and things like that, and made a documentary called Unrest, which actually was in the New Zealand International Film Festival in 2018, a very poignant documentary. And then she co-founded a patient group in the United States called ME Action, which has lobbied for awareness for the illness, for funding into research. And this has really led in 2019 to the US Senate. They weren't able to impeach Trump, but they were able to pass a resolution acknowledging ME as a serious illness which had been neglected in the US and the need for further research. There was only one clause missing, I think, in the Senate Act pass, and that was that people who were doing research on this deserved Nobel Prizes. But okay, so what I wanted to do here is just tell you about my own first meeting with this illness. My 14-year-old daughter, who was highly interactive into everything, very successful in what she did, totally intimidated me. After quite a difficult attack of glandular fever, that's the Epstein-Barr virus, a month or two later, her health just dramatically down-spiraled such that she could only walk for about 5 or 10 meters without having to lay down and rest for 2 or 3 hours. She became allergic to all of her food, so my wife had to kind of take fundamental food items like flour and work out was there a flour that she could take without getting an allergy. She was a bright young creative writer, very talented. She was very good at maths, but she lost those cognitive abilities. And it was the first time I heard the word brain fog. She said, my brain is in a fog. And the other dramatic, apart from lack of sleep and pain, the other dramatic thing was this hypersensitivity to light and sound and touch and smell. And so it was quite hard for me as a Kiwi male in the weekend. If I tried to bang in a nail, it was like torture to her. So I couldn't do that for the 3 or 4 years of her acute phase. So this is pretty traumatic for a family. But in the moments that I could step back from that as a biochemist, I thought, the fatigue was so pervasive, and I wanted to see if we could actually look into what was going on. And we had an academic in the department, Mike Murphy, is now professor at Cambridge, who was working on mammalian mitochondrial complexes and energy production. So we put in a grant to the Health Research Council and got back reviews which said this disease doesn't exist. So it was impossible to do anything at that time. And it took 22 years before I saw an opportunity to come back and actually do some research. And when I looked at what was then in the literature, it was typical research that I would have done as a biomedical scientist. Very focused. People had their own views of what's going on, and so they were looking at some very narrow piece of work. And so it was like sort of very attractive jigsaw pieces, but when you looked at it, you couldn't see what the illness was all about. You couldn't integrate it. So I set a goal for myself of seeing if we could take a much broader approach and try and integrate these pieces and really solve the jigsaw puzzle. Now, as a parent of a ME family, of course, I had on my mind some really high-level questions. You know, what was causing this myriad of very debilitating symptoms I've talked about? You know, there had to be some control center in the body, and surely that was the brain. And why didn't my daughter get better? I mean, so what is it that's preventing the healing process with people with ME/CFS? And then when she moved from the chronic phase to the acute phase after about 3 or 4 years, she was getting frequent relapses, and this is very common among all of the university students that are in my patient groups at the moment, constant relapses. And that can be because of physical or emotional or psychological stress, or it could even be because we've left the door open and there's a cold draft. That was sufficient to actually put them down into a relapse. And then a new question came on the 25th anniversary of my daughter's illness when she was 39, so she was 14 originally. And she rang me up one day and said, guess what, Dad? And I didn't dare say, because I knew she desperately wanted to have a child and she was pregnant. And so that year was very exciting, not just because I was going to be a grandfather, but also because her health improved markedly during the year. And the exciting thing about this for me was that this disease is not irreversible. It's possible, if you found the right physiological state, to really at least make a huge improvement in the quality of life for people. So, you know, of course, one speculates, is it because ME people have low blood volume? Is it because there's an increase in blood volume? Is it because the immune system is suppressed? Or is it because there's more stability in the hormones? And who knows that? But one thing I can say for her was because of this, and that's my grandchild who at 8 months, and he lives in the Waitakeries in West Auckland. And at that time, I just wanted to make sure his future was assured. So you can see that. He's now 5. This is him on his 5th birthday in his kayak, and he's a very healthy young child. My daughter, her health was better, not as good as during pregnancy when she was breastfeeding, but when she stopped breastfeeding, there was quite a very hard fall, and her health's gone back to a lower level status. So the understanding of the disease in New Zealand through this time, I discovered, you know, wasn't great in mainstream medicine, but there was one shining light, and this was a GP on the North Shore called Rosamund Vellings. She was in London during the Royal Free Hospital epidemic as a young woman, and so she was aware of that. And so she came to New Zealand and had a GP practice and had 1 or 2 patients come to see her, which she identified as possibly having ME/CFS. And so in the last 20 years, she's actually, her whole practice is devoted to people with ME/CFS. She's seen about 5,000 patients. and she's written 4 books. The last one on the left there is a revised edition of her first book. And she's been on international bodies drafting guidelines and things. So we're very fortunate to have her as a world expert. And she's been our clinical collaborator and absolutely essential for us to be able to do the studies we've done, because she diagnoses all of our patients either directly or affirms their diagnosis if they've been made by someone else. I also want to acknowledge my very loyal research group that have been working on this, particularly in the last 3 or 4 years when we've resolved a lot of our studies I'm going to talk about. In the middle is my technician who I employed in 1980, and she's the glue that holds everybody together. On the right is Dr. Erin Sweetman. She came to me as an honours student just as we were starting this work and said, I'm going on my OE, scientific OE at the end of the year, and I said, okay. And then at the 11th hour said, would you mind if I did a master's? And then once I'd finished critiquing her master's thesis, she came to me and said, would you support me if I switched to a PhD? And then she stayed on as a postdoc. But sadly, she left us a couple of months ago for her scientific OE across Cook Strait, as her junior doctor partner had a transfer to Upper Hutt. And in between those two is Amber Hillowell, who came and did a genetics honours student and then did a master's and has done an epigenetic study, which I'll talk about. And she's currently employed as an assistant research fellow. She's given fantastic service, a great person to have in the group. And then, because I teach to third-year medical students, I've had medical students come and ask about working on the project. So on the left is Emily Wood, who came as a fourth-year medical student to do her research here. And then on the right hand, there are 2 people who've actually got ME. Angus Mackay, before he had his illness, was a high-performing person. He was in the Scotland under-21 rugby team, along with Gavin Hastings, for those rugby buffs, an icon of Scottish rugby. He was in the British Army and then taught, and then came down with ME, came to New Zealand and asked if he could come down and do a PhD. But he was having frequent relapses, so suspended his PhD, but we've spent many Friday afternoons talking together and developing ideas, and I'll talk about that next. And then down on the bottom, Anna Blair is one of our study group people, and she completed her degree in the middle of the year this year, having been delayed a semester by her illness, and she came and asked if she could do work experience, and she might stay with us now for the next couple of years. So this has been an incredibly loyal group. Sometimes when we haven't been absolutely sure whether we're going to have money to keep going with this project, in a month or 2, they've all hung tight, and I'm very grateful to what they've, how they've supported me and the work, really talented work they've done. So I want to talk just a little bit about what Angus and I have done. I was interested, of course, in these big questions and trying to find a paradigm where we could explain all of the myriad of symptoms, why people didn't get better, and also reasons for the relapses. Angus, on his part, every time he had a relapse, he would hold his head and say, his brain was inflamed, so he believed neuroinflammation was the cause and the solution. And so he was very excited when he found a paper that was published in 2014 from a Japanese group, which had done a PET study on activated microglia in ME patients. And what they showed was that there were activated microglia throughout the limbic system, and there was more evidence of inflammation if the severity of the ME was greater. So we developed this paradigm focused on the hypothalamus and particularly the stress center within it, within the paraventricular nucleus. And really, I've just sort of put it in a nutshell there, that there are cycles of fluctuating chronic neuroinflammation that can drive stress-related relapses in CFS, support the failure to recover the normal homeostatic set point that would lead to recovery. And we published that in December of 2018, and it's received quite a lot of interest internationally. So we were both invited to an international think tank a couple of months ago based in the United States. Now, one thing I realized when we attempted again to get funding to work on ME/CFS from the mainstream investment agencies and failed, that what we had to do was to be very disciplined and focused in our approach. Interestingly, the Ministry of Health had a portfolio of research on long-term diseases, and I thought this is great because ME/CFS would actually fit into that. And the senior official at the ministry told me they would never give money for ME/CFS unless I could show that it was relevant to a disease like diabetes or something. And that's despite the fact that the number of sufferers in New Zealand is estimated to be somewhere between 20 and 25,000. So what we decided to do was to use a precision medicine approach where we would study a small group of patients in great depth, and we'd combine the clinical information that Rosamund Vellings could get from her diagnosis and her following of the patients with biochemistry, physiology, and molecular biology studies to see if we could identify disease patterns and certainly lead to better patient management. For my part, I wanted to really see if I could determine a biological basis for the disease. The other thing, which is clearly a big deficit, is there's no diagnostic tool based on molecular biomarkers, and so we wanted to see if we could do deal with that. So our classic cohort then, it's supposed to be 10 and 10, although I'm showing you one that we used recently, and I discovered there were 11 patients and 9 controls, and don't ask me why that was, but normally it's 10 and 10. The median age for this group for ME/CFS patients is 43, and the average time that they'd had the disease was 11 years. So we're looking at people whose onset of illness was about 30 and predominantly female, and that's what, so it's representative of this group of patients. 9 of them were in the chronic phase, 2 in the acute, and that fits with the kind of ratios that one might expect. And when we asked them to say what the trigger for their illness was, 7 of them said acute infection, of which glandular fever was quite common. 1 was surgery, 1 was stress, and 2 were unsure. And I should say that in addition to these infectious disease outbreaks I talked about, we now know a lot of the ME/CFS, which is in the community, are sporadic cases from things like surgery or stress or childhood abuse or exposure to a chemical toxin, and from these lingering infections like glandular fever, which are around all of the time. So the participants, as I say, were diagnosed by Rosamund Vellings using what we call the 2003 Canadian consensus criteria, which were developed by a group of international experts, and so all diagnosed by the same criteria. Now, the first thing we wanted to do was see if we could actually explore the possibility of a diagnostic test. And one thing by the time we started this work in 2012 was that it seemed clear that ME/CFS patients had a chronically activated immune system. So we wanted to look to see if we could find a possible molecular biomarker which changed its physiological state when the innate immune system was activated. And so what we ended up choosing to look at was protein kinase, RNA-activated protein kinase, or PKR. And this is activated by stress or by inflammation or by viral infection. And so what happens, as you can see on the right part of the slide, is that there's an inactive monomer. So this is inactive when the immune system is not activated. It's got an RNA binding domain. So if it's a viral infection, it'll bind to the viral RNA. If it's some sort of internal event like stress inflammation, then there's an endogenous RNA that binds to that. The monomer then forms a dimer and it self-phosphorylates. And so an activated immune system then has a phosphorylated state of the molecule, and the inactive doesn't. So we thought, well, if we could look at the ratio of the phosphorylated form to the non-phosphorylated form, that might actually serve the purpose. So Erin Sweetman in her master's set out to see if we could test this idea. And so we expressed the phosphopeptide along as a fusion protein with a carrier, and also the whole length PKR didn't express very well, so we also put that as a fusion protein. We expressed lots of the protein, purified it, and then antibodies were raised in rabbits. And then we used negative and positive selections, affinity chromatography to highly purify the specific antibodies that were against, firstly, the phosphopeptide in the row above, and then secondly, in the whole length molecule. And the negative selections were taking out, for example, antibodies against the fusion part of the protein, and in the case of the PKR itself, any antibodies which were against the peptide where the phosphate molecule was on. And so then we used our 10-patient, in this case, 10 control cohort, just to test whether, in fact, these antibodies could detect ME/CFS patients versus controls. So isolated the protein from immune cells, peripheral blood mononuclear cells, and also from neutrophils. And in the controls, we could hardly see any phosphopeptide at all, just a shadow band on a northern, whereas with the patients, you could definitely see the phospho protein. And so when we compared that, even with just 10 patients and 10 controls, it almost was to significance. You can see the p-value of 0.057. Neutrophils there wasn't such a increase in the phospho protein, but it was still trending towards significance, and we published that last year in Diagnostics. So the next step to develop a test that we were hoping to go immediately was to use a larger patient cohort and actually get people that were in the acute phase or just about when a diagnosis was being made. To do that, we would need to go to Rosamund Velling's practice in Auckland, and it would mean doing all of the blood collection and processing in Auckland. So there's certain sort of resource and, some difficulties with that, but we're planning to do that. And then, as sometimes happens in research, things in the laboratory actually have slowed us down, and that is that these highly purified antibodies we prepared and that were active for a while, we noticed were quite rapidly losing activity on storage. And I have to say I'm really frustrated by this, but we had a visit to Dunedin from Steve Redmond, from Australian neuroscientist, and he said to me that he'd been working on a phosphoprotein with highly purified antibodies and had exactly the same phenomenon, and said they found that just going back and not purifying the antibodies as much actually solved the problem. So we're hoping to get to go back and develop antibodies and use the less high purified ones to see if we can move this forward. And the idea and the goal would be to adapt this to an ELISA format, which could immediately move into a ChemPath lab and be available, obviously, for all practitioners to use, and therefore all patients. I thought probably while we're working with this, somebody would come up with a diagnostic test in the international arena, and there has been one that's been touted, and that comes from the Professor Warren Tate at Stanford, who's head of the Genome Center there. He's got a son who's got the severest case of ME/CFS I've ever heard of. He lives in the dark, can't sleep, can't talk, has no visitors, has incredibly hypersensitivity to light and noise and things like that. In fact, he's only had one visitor apart from his family and medical people, and that's Jennifer Brea actually went to see him and wrote a blog about that visit. Anyway, what Ron Davis has done is developed a nanoneedle test whereby you can just put a bit of serum into the, put a bit of blood into the instrument and some salt to stress the blood cells, and all of the cells from ME patients actually showed a change in electrical impedance, a measure of electrical resistance, where none of the healthy controls he used have shown that. So far, what they haven't done is to show whether in other diseases the blood, acts as though they were healthy controls. But there's been a lot of publicity about this as a potential diagnostic test. So most of our goals then in terms of our molecular studies was based around a very simple starting protocol, which was just to take 20 mils of blood from the patients in our cohorts. Tina is fractionated many samples over the last 3 or 4 years, and we fractionate that on 5-fold gradients. She takes the plasma off, peripheral blood mononuclear cells in the middle, and we isolate the neutrophils from the lower layer, from the red blood cell layer subsequently. From the plasma, we've done cytokine analysis and microRNA analysis. The cytokines, we did show some changes in inflammatory cytokines, and then we've been part of an international study, and what has come out of that is that actually the cytokine profiles change with the course of the illness. I'm not going to say anything more about that because it's a very complex situation. The microRNA, a year or two before we started this, it became quite clear that these tiny RNAs, which are incredibly important for regulating our physiology when they're in plasma, they can reflect what's going on in different parts of the body. So we've done an analysis of that and found some microRNAs that were changed, and we've been able to link that up with the cytokines and connecting into pain and connecting into inflammation. But again, I won't say any more about that. What I want to focus on is just to give you some highlights from our major studies on expressed genes, the transcriptome analysis from the immune cells, proteome analysis, the activity of the mitochondria in energy production, and Amber's study on the dynamic genetic code, the epigenetic code, to get an idea then of the underlying changes that are occurring in our molecular biology of these patients. So the first study was part of Erin Sweetman's PhD thesis, and so she did a transcriptome analysis, looked at 13,000 transcripts, and because we've only got the small cohorts, we use quite high stringency in terms of significance values and fold changes. And with that, we found 27 gene transcripts which were increased and 6 that were decreased. And the top 3, it turned out, were all involved in inflammation. There was a pro-inflammatory cytokine interleukin 8, and there were 2 transcripts of encoding proteins which were anti-inflammatory. In fact, they're inhibitors of the tumor necrosis factor-induced NF-kappa B activation pathway. So it looks like this is just giving us a window that inflammation is occurring, but the body's trying to actually enhance its anti-inflammatory ability to counter this. And the right-hand side just shows that we've validated those 3 by quantitative PCR, and the bottom one shows the individual patient and individual control values. 2 of them were to significance, 1 was very close to significance. But we also wanted to look at what the other transcripts that had changed were doing. So we looked at all 33, the 27 up, the 6 down, by functional network analysis, and we're able to show there were lots of functional interactions. And then with ingenuity pathway analysis, we loosened the p-value a bit so that we could actually have more possibilities of having more than one member involved in a pathway that might be identified. And as a result of that, there were some quite clear, significant differences between the patients and controls in immune inflammatory pathways, the cellular stress response and oxidative stress, uniquely not been found before in the circadian clock function. And of course, that relates to, sleep disability and energy control, and also hunger. So that was quite an important finding. We also showed that metabolism was affected, and generally it suggested there was a lowering of the rate of metabolism, and I'll come back to that in a minute. And we also showed there was dysfunctions in the mitochondria and energy production. And again, we've published that last year, and we're gratified that it was the most accessed paper for the year in the journal. So for her postdoc, Erin has done a mass spec analysis, used the SWOT-MS, sequential acquisition of all theoretical fragment ion spectrum mass spectrometry. And so simply what she did was to isolate the proteins from the immune cells, digest them with trypsin, and then firstly you make a library by taking small samples from each of the controls and each of the patients, pooling them, and doing a shotgun mass spec to determine the number of proteins that were represented, and there was about 3,000, so that was her library. And then the actual swath analysis then looked at each individual sample and compared against the library. This is, of course, produces a huge amount of data, and so we then did a principal component analysis to see if we could compress the data into components and see if we could separate the groups. And so the red dots here are the patients and the blue are the controls, and you can see there is a clear separation of most of the patients from the controls. And so we had Mikkel DeLonghi, biostatistician, come and help us, and he did a linear regression analysis and said that statistically we could actually separate 9 of the 11 patients with the 9 controls. So our 11-9 cohort here became a 9-9. And so we compared then the proteins we compared with 11 to 9, but we compared specifically then, we pursued the 9 to 9 and had quite a dramatic finding, and that is that many mitochondrial proteins were up or down regulated. So mitochondria, of course, there are the structural proteins. So you find proteins, of course, in the cristae and whatever. We have metabolic proteins and things like the citric acid cycle, which feed in to the electron transport chain, which is going to at the end produce the energy, ATP. So these are leading into these complexes where the high energy reduced coenzymes, the electrons are withdrawn and passaged along here, whereas the protons are pushed across the membrane to build up a proton motive force. And eventually in this last one, this flows back, and that provides the energy to get to the ATP. So this was published about 2 months ago. And I just put here these were all of the proteins that we found were changed, not so you have to read all of this, but just to kind of emphasize what the extent was. So on the left here, we have proteins that were changed in the mitochondrion generally, and you can see there's more are upregulated, that's the green arrows, than downregulated. Then we look at the citric acid cycle, and again, the same sort of thing. Lots of proteins that have been upregulated and a few downregulated. Beta oxidation, which takes fatty acids and leads it down to acetyl-CoA to go into the citric acid cycle. A few proteins there upregulated. And then we go to the complexes of the electron transport chain. I showed complex 1, lots of proteins that are upregulated, 1 or 2 in complex 3 and 4. And again, the complex that is involved in the actual synthesis of ATP, again, lots of complexes that are upregulated. So this seems to be an indication that the mitochondria are under stress. They're not able to produce energy perhaps according to need. And so there's an attempt to sort of upregulate the components to see if one can address that finding. And there's some recent work which has implied that actually there's a defect in complex 5 and ATP production. And again, this from an Australian group headed by Paul Fisher, and he found upregulation of complex 1 seemed to be a compensation. So that's consistent with this finding. I want to just take a brief diversion here to talk about a major metabolomic study from Stanford that occurred in the end of 2016. This was regarded as the best piece of research on ME at the time. And they looked at 45 patients, 39 controls, and they examined the metabolites in plasma of biochemical pathways with the most sophisticated equipment, mass spec equipment. And what they found was 80% of 612 metabolites that were identified for 64 biochemical pathways were low, suggesting hypometabolism in 20 biochemical pathways. And so the lead author, Robert Nevio, he suggested that what was happening in ME/CFS is the people were kind of like in a state of hibernation where they were operating at a very low level, and this is where a lot of the problems arose. He's contacted me subsequently once we'd published our mitochondrial paper because he's developed this concept further and believes that mitochondria act as sensitive of danger of environmental threats. And he's postulating that there is a cell danger response center in the hypothalamus, which essentially shields cells from further injury when there's some sort of major stress event, like an infectious disease, and prevents further injury and allows healing to begin. But however, if this is persistently recording an environmental stress, then he's proposing that chronic illness results like ME/CFS. Now, this can relate, I think, to our proposal around the paraventricular nucleus stress center, which seems to be important for maintaining illness and causing the stresses. So maybe the chronic neuroinflammation is the signal which is keeping the illness going. Now, I want to just talk briefly, because having got our mitochondrial data, we wanted to look at what was happening in the mitochondrial functions of the mitochondria, in particular the steps leading to energy production. So this was what I was really wanting to do back in 1990 when I tried to get a grant. But since then, an analyzer called the Seahorse analyzer has come about, and we got this delivered to the university in 2018. It's housed in biochemistry. And this allows you to look at the functions of the individual steps of the mitochondria in live cells. So we took the live cells from our patients and we looked at what was happening in each of the steps. And so in here you can see this allows you to look, for example, through oxygen consumption rate, initially at basal respiration, then you can add a drug which will inhibit ATP production, so you can find out from the difference what's the rate of ATP production. Then you can uncouple ATP production from respiratory activity, and so that gives you the spare respiratory capacity, and there's a little blue bar under there which talks about proton leak. In other words, you're pushing the protons to one side of the membrane, holding them there so they can flow back through complex 5 to produce energy. But if there's leakage, then that can lessen the functional capacity. So we took our most severely ill patient in our group, who's bedbound, unfortunately, so an acute phase bedbound, and we looked at this, and that rather strange kind of profile we're seeing here is where we're measuring each of the functions. So we initially have the basal respiratory rate, and then we inhibit ATP production, that goes down, and then we release the coupling, and so now we get spare respiratory capacity. And the top two profiles are two age-matched controls against our patient. And what you can see is that the patient had a lower respiratory, basal respiratory rate, not too much ATP production capacity, certainly very little spare respiratory capacity compared with the controls. And one thing about when we are stressed, we want our mitochondria to be able to respond to use that spare respiratory capacity. So if ME people don't have this, then obviously they're not able to respond. I might say this was the first experiment we did, and this is a case where you shouldn't do the experiment twice, but maybe we learnt quite a bit from that, and that is that when we actually looked at this patient's profile again from subsequent studies, subsequent blood taken at subsequent times, in some of them he looks quite normal and in others he doesn't. So this might be a dynamic thing in patients where from time to time their capacity to produce energy is very compromised, but it can actually come back to normality at other times. And so we've got sort of more work to do on that. There's been an index called the Bioenergetic Health Index defined, where if you have good mitochondrial health, you can measure all of these things we've been measuring and get a high Bioenergetic Health Index number. Whereas if perhaps with ME you have rather poor function, for example, you can see the yellow bar here is quite high. That means more proton leak, not too much ATP synthesis, and you can see not much for spare respiratory capacity shown in green here, then your BHO would be low. So we applied that, Emily applied that in a small study with our patients and our controls, and you can see a difference, but surprisingly some of the controls were quite low as well. And so we're doubting whether this could be used as a diagnostic tool, although subsequently Tina's gone and looked at a number of controls. We thought they would be in a tight bunch, and this latest series, they all seem to be actually much more the same. So again, some more work to be done in that. So the last thing I want to talk about is Amber's study. From what we had seen then up to this point was that there were examples of alterations in rates of metabolism. We saw some genes whose expression was upregulated and downregulated. So we wanted to actually go back to the DNA and see, could we explain this by the dynamic code that was been discovered now for 10 or 15 years, the epigenetic code, where there are small tags put on cytosine residues and taken off, and they can be responsible for controlling the levels of expressions of genes. So Amber took isolated DNA from 10 patients and 10 controls, and then she used those to generate reduced genome DNA methylation maps. That's looking at millions of sites across the whole genome using reduced representation by sulfite sequencing. And this is a collaboration with Aniruddha Chatterjee's group in the pathology department, because he's now a world expert in epigenetic analysis. And that was a very happy and fruitful collaboration. So what Amber found was identified regulatory clusters for 17 protein encoding genes related to metabolic and immune activity. Within the gene bodies, 122 genes were affected, and as yet the theory on what it means when we methylate within the gene bodies or take methyl groups off, the theory hasn't really kind of formed on that. So we're waiting for the theory to tell us exactly what this means, but we certainly found both hyper and hypermethylation. In other words, methyl groups have been taken off compared with controls or added on. The important thing from a neuroscience point of view about this illness is that we found from this a number of neurotransmitter and neuropeptide pathways, which was indicating a disturbed neurological pathophysiology, more so than we'd found from the other studies. And we're excited that this was accepted for publication a couple of weeks ago. And so critical finding from Amber's study was that we found 5 genes that were hypomethylated, that is, they'd lost methyl groups within the gene regions. And one of them is a GABA receptor, and one's a calcium channel, and 2 are guanine nucleotide binding transmembrane proteins, and the 3rd one is a regulator of neurotransmitter release. So this can impinge on these transmitters, and we can then relate this to effects on the hypothalamus and the paraventricular nucleus, and what I referred to at the beginning of dysregulation of the HPA axis, ultimately producing the stress hormone of the body, cortisol, which is low in ME/CFS, and that's associated with fatigue. And just a recent study has shown that a unique characteristic of ME/CFS is an inability to repeat an exercise, say 24 hours after doing it. So if you test ME people against healthy controls, they do quite well in initial exercise, but 24 hours later, they can't function in performing exercise in anywhere the same way. So that's called post-exertional malaise. Sometimes leads to severe relapse. You can see this student is asleep on her computer. We're doing, in the last 2 weeks, we've actually done an exercise physiology component of a post-exertional malaise study, and we're going to do a lot of molecular studies on this next year. So just to sum up then, what we've been able to find from all of the molecular studies, certainly relating to neurological affected pathways in ME/CFS, is that certainly there seems to be a dysregulation of specific neural functions affecting the HPA axis. There's changes in the proteins of the circadian clock, which obviously controls circadian rhythms involving sleep and energy and hunger. Certainly effects on changes that promote inflammation as well as trying to control it. We've seen changes in mitochondrial function and regulation, strong evidence of hypometabolism and underperforming metabolic pathways, and overarching that, of course, is in every study we've done, we've seen changes in functions in the regulation of immune cells. Okay, just to the last thing I would like to talk about is just briefly on how does this relate to COVID-19. Well, the surprise finding to some who've been involved in looking after these patients is that quite a high proportion are not recovering as expected, say 2 to 3 weeks after they've had their initial infection. And now, of course, from the first wave, we're more than 6 months past that. And so it looks like that COVID-19 is going to be a generator of a whole new raft of ME/CFS patients. You may have seen in the Sunday Star Times yesterday there was a profile of a New Zealander who had a mild case of the disease. She was a marathon runner, and she's now been recovering for 8 months, and she's got classic symptoms of ME. She's got fatigue, she's got brain fog, sleeplessness, just the kind of things which ME people have. So there's an estimate about 30% of cases haven't recovered, and so a proportion of those may indeed go on to get ME/CFS. That would probably double the estimated world incidence of the disease from 15 to 30 million. I mean, so far we've had, I think it's 60 million cases now in the world. And one kind of slightly positive note is that because these are just these people are going into a possible illness, it might be possible to early intervene with some developing therapies. And we've been working with antioxidants with mature ME/CFS patients and had some evidence that antioxidants might be helpful. And so there is a body of feeling, certainly, and people are publishing papers on that, promoting antioxidants like N-acetylcysteine, coenzyme Q10, MitoQ, for example, that might actually be helpful. But time will tell. So I'd like to acknowledge then, people that have enabled us to actually do all of these studies. We've had financial support when possible from the national body NZMEs, and obviously support for us to do the studies. And we've had the amazing difference that's been made, and why we've been able to keep going and keep a group together and do them was because private individuals and families affected by this illness have given us incredibly generous donations, and often repeated ones. We've had some grants, modest grants from Lotteries Health and also Otago type charities, but as I said before, not managed to get funding from major investment agencies for this. And of course, we're very grateful to all of our ME/CFS study participants. For the post-exertional malaise study we've just been doing, young woman students stayed behind after their exams to put themselves through a rigorous exercise regime, and we held our breath and hoped that they would go off and not actually go into a relapse as a result of that. And so far, I think everyone's okay. So thanks very much for your attention, and I hope I've shown you that actually this is an interesting disease, but it's certainly got a biological basis. Thank you.
Thank you very much, Warren. I'm just wondering, in terms of time, whether there might be anyone on Zoom that wants to ask a question. But in the meantime, is anyone in the audience got a question for Warren.
Yes.
Very much. That was really interesting. Right at the beginning, you mentioned that some of these patients have got therapeutic drug intolerance. Do you mean an increase in side effects or a decrease in efficacy? And depending on which one, do any of your biochemical mechanisms?
Yeah. They're very sensitive to when they take drugs, you know. Certainly generally have to take much lower doses to be able to not get all sorts of side effects. So that's what I mean by intolerance, actually. And so, like, you know, what you find, and certainly in my daughter's case, she tries not to take anything. I mean, when she had her baby, she didn't have any medication, had 48 hours of labour with no medication at all. So, I mean, you know, and many people are doing that because they just suffer so much from taking the drugs.
Are there any model systems? Yes?
Any
model systems?
No, I wish there were animal models of chronic fatigue, or maybe the minks in Denmark might be feeling a bit unhappy at the moment. No, I mean, that's, you know, that's a difficult thing when there's no obvious model, except, well, there is in the sense that, sorry, I'm coming from another point now, the model of hibernation in animals, for example, is that they are lowering their metabolism to essentially go through that. There's torpor in rodents, for example. When rodents feel danger, they actually stay in one place and they can lower their body temperature and you know, they shake a bit. And so just recently, in fact, there have been 2 neurological circuits been found. I mean, 2 separate groups claim they found neurological circuits in the hypothalamus. One is called the torpor center and the other one maybe the hibernation center. They may be the same thing, I don't know. So that could be an animal model, at least for this, you know, danger response center or the cluster of neurons in the paraventricular nucleus, which might be driving the illness, okay? Yeah, but we don't know whether these centers exist in the human brain yet, but that's very recent. So that's the nearest thing. So thanks for that question. Yeah, sorry.
I just wanted to follow up on you said that during pregnancy she felt much better.
Yeah.
And also during breastfeeding. Has anyone looked at that and like what's the biological mechanism there? And is it related, for example, to prolactin?
Yeah. It's a really good question. When I was in London in 2017, I gave a talk to patients, and there were a lot of mothers and daughters there and practitioners, and my daughter gave me permission to talk about this, and everyone was really excited. I thought this would have been well known, but it didn't seem to be. And in fact, a lot of mothers and daughters came up and they were asking about it. I've never seen so many mothers that wanted their daughters to get pregnant. But the critical thing is I've looked at this, and now that there have been studies looking at the effect of pregnancy, and unfortunately falls into 3 groups. About a third of them people seem to get benefit, a third are much worse, and a third have no change. And so maybe if you could separate into those groupings. I was asked, are we going to do any studies on this? And I said, I'll go back and talk to the woman members of my group and see whether in fact they want to. I couldn't actually say we were going to do studies, but it is an interesting question, and certainly, I mean, as I said, really exciting to see the symptoms really, you know, depress markedly, because, you know, that says something about the disease. We just have to find the triggers really to hit the switches.
Could I please ask a question? Hi, my name is Charm. I'm an Otago Medical School alumni, and I have been affected by ME for the past 10 years or so.
Yes, we can hear you.
Oh, okay. I was just wondering, what are your thoughts regarding transient receptor potential cation channel subfamily and member 3? I recently read a study speculating that the cause for ME/CFS may be that this gene is faulty and that TRPM3 of the faulty expression of this receptor may be the cause for this illness?
Yeah, I can comment on that. I mean, this is from an Australian group and they do very nice work, but it is very focused work. And so they've found this channel is calcium channel is not functioning. The data that there is a polymorphism in it in ME people is, I think, much weaker, and so it may contribute significantly to the disease. Maybe one of the things that contributes significantly, but you know, as I say, I respect the work, but I don't think it's the magic bullet. You know, that's my conclusion.
Oh, thank you.
Thanks. I just wanted to ask, you mentioned antioxidants being taken as a possible early intervention with this COVID statistics that are going on. You said some of the people who've had ME for a long time have tried it.
Yeah.
I just asked what that specifically is.
Yeah, sure. So let me talk about N-acetylcysteine first. N-acetylcysteine is a precursor to glutathione, the antioxidant in the body, which is at the highest concentration. So I heard about this first when I was working on HIV, and a colleague said, a group in San Francisco were working, and they were giving HIV patients N-acetylcysteine because it actually stimulated the proliferation of T cells, and of course, HIV T cell levels go down. And there were 2 or 3 people with ME actually in the greater research group. And so they tried this and claimed that they got some benefit from taking this precursor to glutathione. So, you know, we've got information now certainly that oxidative stress seems to be important, and we're going to do more work on this next year. So you could imagine that you can understand a biological basis where N-acetylcysteine might be helpful. I came back and we got some for my daughter, and she took it for a while, and it's hard. It's like many things with ME/CFS. It's hard to know whether it had benefit or not. That's the difficulty. And so that's the reason that N-acetylcysteine is being suggested. MitoQ, of course, is a targeted drug to mitochondria. So coenzyme Q10 with a little tail on, which targets it to mitochondria. And we've done studies with our patients, both in vivo and in vitro with their cells. We can certainly see it going up into their cells when we incubate it. So we can see the targeting occurring. We've done one study with an ME patient, university student, where we got her to start MitoQ, and we took blood samples for 5 months. And what we showed that by month 2, her mitochondrial functional profile had improved, and then it stayed stable and then suddenly dropped. And when I talked to her about what happened, was anything happening between month 4 and month 5, turned out she had surgery in that time. So it's only an N=1 study, but we did seem to get some improvement with MitoQ. The difficulty is when you asked her, did you feel different? Subjectively, people have not been able to say that they felt different, even though biologically we might have found there's some improvement. So that's the difficulty, really. But it has, a recent paper published has suggested that MitoQ should be used because mitochondria now are thought not only just to be organelles which make energy, but they seem to be these sensors of things going wrong in the body and maybe importantly in the brain. So when there's some dysregulation in mitochondria, that seems to be sensed as a stress and a danger. And it may be, that could be an important reason to take an antioxidant which kind of restores mitochondria to a more normal state. Does that sort of answer your question?
Okay, I think we better wrap things up, but I'm sure Warren's happy to sit and discuss further about his research. In the meantime, we have afternoon tea outside, and then the poster session continues. We'll come back about 4:10 for the prize giving, which includes both the prizes for posters but also some other awards. So I'd just like to finish by thanking Warren for a fascinating talk. It really illustrates really well how biological this disease is, and it's not just a figment of someone's imagination. And hopefully, in the near future, you'll get some major grant funding for this. So I'd like to present you with a token of our appreciation.
Thank you.
And also a drink bottle with our new BHRC logo on it.
Here we go.